All right, so introduction to biochemistry. Learning objectives, we're going to be able to, or I want you to be able to describe the structural features for each of the biomolecules and use the proper nomenclature to determine them, or to describe them, sorry. Draw a covalent linkage between two amino acids to make a peptide and describe the characteristics of this linkage. Draw how the different components of nucleotides are linked together. Draw how monosaccharides are linked together to make other polysaccharides. Describe the thermodynamic terms enthalpy, entropy, and free energy in chemical processes. Know their symbols and demonstrate how they relate to equilibrium and the spontaneity of a reaction or process. Draw an energy diagram and understand the different parts of the diagram and what they tell you. List the differences and similarities between eukaryotes and prokaryotes. Describe the role for each of the various eukaryotic and prokaryotic cellular structures, the nucleus, the flagella, etc. Now, when we get into the building blocks and the biochemistry of living organisms, it's really important that you understand that almost everything in a given cell or in a living organism is a polymer. Now, those polymers, they vary a little bit from one another, but those polymers are the building blocks that are kind of structural elements and other elements that help organisms live. Biomolecules obey the same chemical and physical laws as all molecules. So just because something might be essential for uh, structural integrity of the nucleus or something like that, at the end of the day, it's made up of carbons, hydrogens, nitrogens, oxygens, phosphorus, and sulfur. And so, being made up of those things, they're going to behave in the exact same way. So, I think that it's important to go ahead and recognize that a biomolecule isn't really anything special. A biomolecule is a molecule that um, is it just happens to be a part of a living organism. At the end of the day, it's a molecule with the same physical properties as some sort of synthetic or synthetic or inorganic molecule. Oxygen, whether it's in a protein, a nucleic acid, or um, a molecule of ozone, it's going to have the exact same properties and it's going to behave the same way. It's still going to be an atom with an electronegativity of above 3. It's still going to have basically a desire to get 2 additional electrons to it. So that's one thing that I think is really important to remember. Biomolecules aren't some special thing. They're just like every other molecule. Now, our examples of our biomolecules are proteins. Proteins are kind of the agents of change and metabolic flux within living systems. So that's one class of biomolecules. Another are polysaccharides. So these are sugars. Now, this polysaccharides also includes saccharides, whether they're mono or disaccharides. Sugars essentially is our next grouping. Then it's nucleic acids, which contain the genetic material of an organism. And then last but not least of our major biomolecules are lipids and membranes. So our lipids and membranes, they're the only one of these four that are not going to be a polymer. Now, lipids do kind of aggregate together and they do form molecules that are larger than just one single fatty acid, for example. But lipids are not going to be the repeating units that go on and on forever and ever, as is the case with our nucleic acids, potentially some polysaccharides and proteins. The next separate category of biomolecules is our metabolites. Now, metabolites are molecules that basically their primary role is to extract energy from them. And one example of a metabolite would be something like glucose. Now, what you see here on the left-hand side of the screen is essentially one of your major building blocks for a biomolecule. So I'm going to circle this. This right here is an amino acid. Now, that term we're going to dig deeper into later on, but I want you to know at this point we've got amino group and a carboxylic acid group, hence amino acid. So every single amino acid is going to have that. Every single amino acid is also going to have a singular hydrogen coming off of a central carbon. This central carbon is known as your alpha carbon. Highlighted that red. Now, you've probably heard that there's proteinogenic amino acids. There's about 20 proteinogenic amino acids, 20 or 22, depending on what source you look at. And this group right here, this R group, that's where your variation for every proteinogenic amino acid is going to be found. So every amino acid has 4 different groups coming off that central carbon. And when you compare every amino acid, 3 of those 4 groups are identical. The 4th one is a little bit different, and that's what I highlighted in green there. Now, like I was saying on the previous slide, every protein is made up of these amino acids. So every protein is a polymer. Now, if you look at this group circled in, or this molecule circled in yellow, what I want you to do is track with me the atoms. I'm going to go ahead and clear all my erasing, or clear all my markings. And I'm going to highlight groups on this model, and then on my polymer model shown below. Okay, so this NH3 plus, oh, I didn't highlight it, it just kind of erased it. So highlight, I'll do it in yellow. That NH3 plus right there, that group is this group. Now that central carbon right there, that is this right here. Okay, now off that central carbon, what you'll notice is you'll have on the right-hand side that hydrogen. I'm going to highlight that and that. You'll also have your R group, basically pointed downward from my chain. And then you'll have this C double bonded O, and that is that right there, that C double bonded O. Now, after that group, obviously I don't have a carboxylate ion, but what I do have is a C to N bond. So that's a covalent bond, carbon to nitrogen bond. It's a covalent bond, and that's not depicted here. Why not? Well, the reason is that we're looking at the individual subunit on the upper left-hand corner, and then right here, I'm looking at my polymer. So that carbon and nitrogen is this carbon on the top, so this one right here. But what I've done is I've lost an oxygen. So I have a carbon-nitrogen and then a nitrogen-carbon, which, oh, lo and behold, I've restarted this process, or I've restarted this molecule shown above. So now I'm going to switch my color again. I'm going to switch to orange. This carbon, or sorry, this nitrogen is another hypothetical amino acid. That carbon is right there. And then I've got my carboxylate ion. I've got my carboxylate ion. Okay, so this is 2 pieces of or 2 amino acids put together to form what's called a dipeptide. We'll get more into that later on, but basically this is the way in which biological molecules are formed into higher order structures. These 2 building blocks get put together and they make something bigger than themselves. And they'll continue doing that until they form something like what is shown below. That molecule that's shown below is this, but rather than having 2 amino acids, it would be, see, I would bet closer to like 150 or something like that, 150 amino acids. Now on the right-hand side of the screen here, we've got an individual sugar. This is a monosaccharide. What I want you to notice about this sugar is we've got a bunch of hydroxide groups, those hydroxide groups can work as decent nucleophiles and they can participate in reactions. Down below, we've got, oh, this group or this molecule that looks a little bit like this molecule that looks a little bit like this molecule, and so on. There's some differences, absolutely, but basically what we have is this is known as what, or this is a polysaccharide made up of repeating units of a sugar. Now, those multiple subunits of that sugar can combine together to make a higher order and more complex molecule, like what's shown here, where we talk about something like cellulose. Cellulose is a structural polysaccharide or structural sugar in plants that basically helps plants stand upright. Now, that is, at the end of the day, just a bunch of carbons, hydrogens, and oxygens. There's some nitrogens in there as well, but we won't worry about that for the time being. So we have these repeating units going on over and over and over again to give us this higher order structure, this cell wall, these cellulose molecules that make a molecule stand up, or make a plant stand up. Now, on the left-hand side here, and on the right-hand side, we got a couple of different examples where we're kind of doing the same thing. Now, on the left-hand side, what I want to draw your attention to is this right here. That right there is basically one side of this molecule, which is shown below, a double-stranded piece of DNA. It's one small fraction, one small subset of this molecule down below. Now, with that said, this molecule on top, I'd like you to recognize a couple of things about it. One thing that I'd like you to recognize is the type of bond that connects the different components of this, because basically what we have is this molecule being found here, and then again being found here, and repeating over and over and over and over again. So how do they repeat over and over and over again? Well, one way is by having this type of molecule, which is known as a phosphodiester linkage, or this type of bond between different groups. This phosphodiester bond connects sugars to one another in the 5' to 3' directionality. So this is a phosphodiester linkage that connects a 3' carbon and a 5' carbon. So you have these molecules that just repeat over and over and over again. Now, this side of the molecule right here, this portion of the molecule right here, what that's going to do is that's going to actually establish hydrogen bonds with the opposite strand of this double-stranded piece of DNA. Now again, we'll get into this a little bit more later when we're dissecting DNA structure, but this is just the thing that I want you to be thoughtful of. Like, be thinking about, okay, I know the different types of bonds, covalent, non-covalent bonds. Okay, but now let's look at those non-covalent bonds. Well, we have not electromagnetic. We have our intermolecular forces that bring molecules together. What are those different intermolecular forces and where are they going to be found? Well, hydrogen bonds are a very, very common intermolecular force that we'll see in this class. Okay, then if we look at the right-hand side of this slide, there's a couple of things that stand out. One is that we have a molecule right here. And then we have kind of basically the cartoon version of it. This molecule is a glycerophospholipid. Now this glycerophospholipid has a phosphate group, has a glycerol backbone, then has some fatty acid chains. Okay, now those fatty acid chains, well, they're very non-polar. They're very hydrophobic. Whereas the head group here, it's a very polar head group. It's a hydrophilic group. Now, these molecules, this is basically as not necessarily as complex as a lipid is going to get because they will aggregate together, but you're not going to have a repeating lipid, something that just goes on in perpetuity. Now, these things are going to be basically your building blocks for something like that we see down here, where we have a fatty acid, or we have this molecule, this blue head corresponds to this blue head right here. These two yellow tails correspond to two yellow tails in here. Um, and that's how we kind of stitch it all together. Another important thing that you need to be thoughtful of and think about with respect to this class is the central dogma of molecular biology. You might also have heard of this as the central dogma of biology or biochemistry. In its classical interpretation, or its simplest interpretation, it was simply DNA goes to RNA, goes to protein. But what we've seen is that that's not really that's not the it's not quite that simple. It's not just the DNA molecules converted to RNA and the RNA ends up being produced as a protein, in part because you have DNA that replicates, you have RNA that acts as an enzyme. RNA can regulate different components of gene expression. So there's a lot more to this. But I want you to be familiar with these terminologies. So translation is the conversion of RNA to DNA to protein. Transcription is DNA to RNA, and then replication is a way of using DNA to make more DNA. Now, structural and stability of biomolecules. Covalent bonds are your strongest types of bonds. Now, you can have multiple ionic interactions that give you a strong ionic bond, but I think that it's helpful to just kind of go in with the mindset of, okay, covalent bonds are my strongest bonds. We've also got non-covalent bonds, or our weak interactions. We've got ionic interactions, electrostatic interactions, hydrogen bonds, van der Waals interactions, and hydrophobic interactions. Now, when it comes to what we will see in this class, I would say that hydrogen bonds and hydrophobic interactions are the ones that we'll talk about specifically most often. Does that mean that ionic interactions or electrostatic interactions don't take place? No, it doesn't. They absolutely do, just as van der Waals or induced dipoles do. But more times than not, when we're talking about, the stability of something like DNA, what you're focused on is you're focused on those hydrogen bonds that hold the two strands of DNA together. Hydrophobic interactions. Well, hydrophobic interactions drive proteins to fold in a certain way. So, ionic interactions, hydrogen bonds, hydrophobic interactions, those are going to be your three most common. And so I think that it's helpful whenever we look at a molecule or when we're looking at like a cross section of a cell, kind of ask yourself, where are my hydrophobic interactions? Um, where are my hydrogen bonds? Where are my ionic interactions? And in many cases, when it comes to hydrophobic interactions, what's important to remember is that we're doing all of this in an aqueous environment. So we're not doing this in like an organic solvent like hexane or anything like that. Instead, no, this is just a solution, or sorry, it is like a molecule that's partially electronegative, or not partially electronegative, partially hydrophobic that is going to aggregate with other partially hydrophobic chunks or parts of that system. Okay, so reaction rates. When we start talking about enzymes, we're going to go deep into reaction rates. And when we see a general rate expression, rate equals K times A, or concentration of A times concentration of B. And whenever you look at chemical system, and when we look at a biochemical system, the same properties are true. Rate is equal to K times the concentration of A times the concentration of B. Um, the rate can be influenced by, an increase in temperature, an increase in pressure. Change something about the environmental conditions, and you're going to change the rate of it. So we also put many different things in the context of thermodynamics, and we're concerned with thermodynamics of different chemical reactions and multiple chemical reactions. As biochemists, we're concerned with the thermodynamics of metabolic pathways as a whole. So not just individual reactions, but a reaction that leads into a reaction, which leads into a reaction, which leads to something else, and so on. So we want to bring back and revisit thermodynamic terms that you learned as far back as Gen Chem 1. Um, so enthalpy. Enthalpy is denoted as delta H. It is the sum of the internal energy and the product of pressure and volume given by the equation H equals U plus PV. Now, when a process occurs at constant pressure, the heat evolved either is released or absorbed, and that is equal to the change in enthalpy. So we use terms like exothermic, endothermic, and isothermic to describe the enthalpy of a reaction. An exothermic reaction is, or an exothermic process is one that releases heat. Now, entropy. Entropy is a thermodynamic property that people always, I remember when I was in high school, it was entropy was described with the example of like your room or your workspace. And well, it observes entropy. And I remember that kind of phrasing was used, but that there's a little bit left to be desired there. So um, a workspace. So the idea is that, when you start out the semester, let's say that your workspace is very organized, but then if you don't on a daily basis pick it up and reorganize it and reposition all of your belongings, then it's going to become very disordered. And at the end of the semester, at the end of some time frame, it's going to be a complete mess. It's gone from very organized and very ordered to extremely disordered. Now, that is an increase in order, or sorry, an increase in disorder. And that is best described as an increase in entropy. So entropy is a system which becomes more disordered. So entropy is denoted as delta S, and delta S, you'll sometimes see that as or so that sometimes is counterintuitive almost, because people will hear, oh, okay, well, it becomes more ordered. That's a good thing. And then you bring in your own personal like feelings about it, and you're like, good is positive. So when something becomes more ordered, that is positive. Well, that may be true, but that's like your opinion and that's your perspective. When we talk about entropy, entropy is a positive value, denotes an increase in disorder. Last but not least is Gibbs free energy, which is denoted by delta G. Now, delta G is the important thermodynamic term. So delta G is going to indicate whether or not a reaction is spontaneous or non-spontaneous. So a negative delta G value will indicate a spontaneous reaction. Now, Gibbs free energy is the total amount of energy available to do work at constant temperature and pressure. A reaction is exergonic and endergonic, or is classified as exergonic and endergonic. Now, when it comes to thermodynamics, you need to be familiar with the laws of thermodynamics. Um, the total energy of a system and its surroundings is constant. The total entropy of a system of a system plus that of its surroundings always increases. So entropy can decrease locally in the system if there is a corresponding increase in entropy in the surroundings. Now, thermodynamics of a reaction. Here what we have is we've got on the right-hand side, we've got short pieces of synthetic DNA or short pieces of DNA that's existing as a single strand. And when you mix those single-stranded pieces of DNA together, what's going to happen eventually is those single-stranded pieces of DNA are going to aggregate together and they're going to basically reform and make a system more ordered. So now this question, does the entropy of the DNA increase or decrease during each of the processes? So when we go from mixing or that first cell to the second cell, what's happened? Well, one way that you could look at it is you've got a handful of blue molecules in this confined space. And then when we remove that barrier and mix them together, now all of a sudden we've got blue molecules occupying this space. So the space went up. Um, and each of those molecules occupies a different space. So that would be an instance where those blue molecules and those pink molecules, by distributing themselves more, the entropy of that system went up. Okay? The disorder in the system went up. Now, the last cell, where we have blue and green molecules together. Okay, so those blue and green molecules mixed together, well, they occupy comparable spaces, but now all of a sudden they've come together. So we have a system that's a little bit more ordered. So that would be an example of my entropy decreasing. So the system became more ordered when I went from, what would that be? 4, 7, 10. That's a total of 20 uh, molecules. Let's say I had 20 molecules. I threw them in this second spot right here. Those 20 molecules are kind of wherever they want. But now down here, my total number of molecules dropped from, well, 20 down to 10. Now, Gibbs free energy is something that, again, we're going to talk about quite a bit when we get to enzymes. So the Gibbs free energy equation, delta G equals delta H minus T delta S. When we have a KEQ that is greater than 1 or a KEQ that's less than 1, I think that it's helpful to think about what would your delta G be there. So if a reaction, if a KEQ is greater than 1, that means the concentration of our products is greater than that of our reactants. So that would mean we should have a delta G that's a negative value because that reaction is naturally going to progress towards my products. Now, for a spontaneous process, when we look at a chemical reaction free energy diagram, we've got our reagents going to our products. And what happened was our free energy was low, then it went up, and then it went lower. So this would be an overall exothermic reaction because our reaction we gave off gave off energy when this reaction was completed. Now, we will not spend a whole lot of time looking at the um, different properties of living systems and living organisms. Um, the three major branches of organismal life are bacterial, eukaryotes, and archaea. Traditionally, eukaryotes and prokaryotes, but this is a little bit more accurate of a explanation. Now, if you look at the top of this figure, what we've got are a couple of different organisms. We got Sulfolobus archaea, Arabidopsis thaliana, and we've got Linus pauling, Homo sapien. Now, these organisms are radically different from one another. All of these organisms undergo replication, transcription, translation, and duplication. Now, these organisms look so radically different. But then if we look at the bottom here, where we have selected or found a Tata binding protein, what you'll notice is that all of these different organisms, their binding proteins are very, very similar to one another. So despite their like kind of phenotypic difference or their difference on a growth scale, at the molecular level, their similarities are very, very impressive. Now, if we look at this model or this from your chapter, your textbook, we've got an animal cell which is approximately 100,000 nanometers. If we compare that to E. coli, E. coli is 15 nanometers. So the differences between these in scales is pretty radical. Now, when it comes to terms that I want you to be familiar with for chapter one, it's all of these: covalent bond, polymer, condensation reactions, proteins, polysaccharides, nucleic acids, lipids, and thermodynamics, equilibrium, enthalpy, entropy, Gibbs free energy, spontaneous reaction, prokaryotes, eukaryotes, and organelles. So look at all of these and make sure that you feel comfortable with all of these different terms. Um, some of them, excuse me, we just kind of touched on lightly. Excuse me. So what I would do if I were you is I would look back at an old textbook and make sure that I feel good about where I'm in these different terms. There's a couple that are going to persist and become kind of more important as we work through this entire semester. One of them is covalent bonds. And the number of times that I've taught this class, um, I think that the types of bonds are something that people have a tendency to kind of take for granted for. Um, and so, give yourself some examples. Like if I see a carbon-carbon bond, I am thinking covalent bond. Carbon-nitrogen bond, I am thinking covalent bond. Because what ends up happening is that people forget kind of your definitions. And one example that I'll give you kind of as a little bit of an aside is hydrogen bonds. Hydrogen needs a total of four different atoms. Hydrogen is one of them. Then nitrogen, oxygen, and fluorine. So it's hydrogen, nitrogen, oxygen, and fluorine. Now my question, and this is kind of an application question, is how often do you see a carbon hydrogen bond that will participate or a carbon atom that will participate in hydrogen bonding? And the answer is you don't see that because that's not what happens. For hydrogen bonding to occur, we have to have nitrogen, oxygen, or fluorine. Those three atoms are super electronegative. Then the other atom that's kind of along for the ride is a hydrogen. All right, well, make sure that you review these concepts. If you could come up with like, just as a little suggestion, like if you had a index card with just like a one quick summary of any and every one of these topics, I think that would be helpful for you to review it. All right, well, have a good one.